Progress of research in life science and tight collaboration in relevant industry chains are essential for achieving better vital quality. Originated in 2002, RWD has been constantly furnishing customers with reliable solutions and services in life science, animal health and clinical medicine, and dedicated for better vital quality within our wits and powers. Our products have been sold to over 100 countries and regions around the globe and won worldwide customer confidence and support. At RWD, we launched 6 solutions in the fields of life sciences, animal health, and clinical medicine at the forefront of the world, including animal surgery and modeling solutions, which ensure experiment efficiency through diverse animal disease modeling and model evaluation techniques. In vivo imaging solutions, which provide tangible tech support in fields such as biology, pharmacology, materiology, and translational medicine, among others, by accurately showing blood flow and changes in other indicators of live beings in visualized imaging. Neurosignal research solutions, which are valuable tools for neurocircuit analysis, as well as research into mechanisms for neural system diseases, development of brain-machine intelligence technology, and more. Histopathology solutions, which improve the quality and efficiency of pathological diagnosis while sectioning with high accuracy and efficiency. Cellular and molecular biology research solutions, which advance efficient scientific research through covering all conventional experiments, including cell separation and purification, self-culturing, self-analysis, and molecular biology. Animal diagnosis and treatment solutions, which evaluate diagnostic efficiency and treatment efficacy of surgery with anesthesia, dental services, rehabilitation, and physiotherapy, urinary system diseases, etc. And our assistants, science researchers, have published thousands of papers in a number of world-leading academic journals, including Nature and Science. On top of that, many scientific research institutions, medical institutions, and pet diagnosis and treatment centers have attained their success by leveraging our 6 solutions up to now. We will maintain cooperation and innovation with an open mind, boosting growth within many industries through joint efforts with our partners.
So we've been collaborating and cooperating with RWD for quite some time now, and their self-developed products have helped us in the laboratory maintain accuracy and improve accuracy and help our efficiency. We've used everything from cryostats for sectioning various organs of the body, all the way through to stereotactic surgery solutions to deliver small amounts of virus to very minute regions of the brain. We're really happy with RWD's products, and we're really excited to see what they've got in store, their future products that they have in store for the future, and helping researchers worldwide.
Mein Name ist Dr. Heinrich Krollebusch. Ich bin seit 30 Jahren praktizierender Tierarzt und seit 20 Jahren beschäftigen wir uns schwerpunktmäßig mit der Narkosetechnik in der Kleintierpraxis. Uns Tierärzte treibt das Wohlergehen unserer Patienten an, sowie die ständige Verbesserung ihrer Lebenserwartung und ihrer Lebensqualität. RWD hilft uns dabei mit hochmoderner Beatmungstechnik und ebenso modernen Lösungen für Diagnostik und Therapie. RWDs Hilfe können wir Tierärzte sicherer und effizienter operieren, aber gleichzeitig sorgt RWD auch für eine verbesserte Lebenserwartung. Das Wohlergehen unserer Patienten und die ständige Verbesserung ihrer Lebenserwartung und Lebensdauer treibt uns Tierärzte an.
As a national high-tech enterprise, RWD ascribes great importance to technology research and development. Based on 4 technical platforms, including optical imaging, precision transmission, accurate temperature control, and detection of weak signal, we have a grip on leading core technologies across many fields. Since RWD's founding, the company has invested hundreds of millions on research and development, gaining almost 100 patents, both domestic and international, as a result. In addition to a sound manufacturing base, advanced processing assembly line, and precision detection system, RWD has built an integrated control system that monitors all aspects of operations from research and development to manufacturing. The name RWD comes from the transliteration of the English word reward, meaning payback to society. Reward is not only the original aspiration of the creation of RWD, but also the continuous belief of the company. In 2016, RWD launched the RWD Preclinical Medicine Enlightenment Scholarship to help with the development of research talent in China's preclinical medicine. So far, the scholarship has been awarded to 38 institutions of higher education nationwide. As the Vice President Unit of the Huxley Wildlife Conservation Society, RWD has donated medical equipment and supplies for wildlife relief and the conservation of the natural ecosystem. In 2020, RWD donated clinical medical supplies worth over RMB 1 million to the Giant Panda National Park to help with safeguarding our national treasure. In the future, RWD will continue to contribute its wits and powers to enhance vital quality and help global customers unlock vital mystery and safeguard vital health.
Hi, everyone from all over the world. Good morning, good afternoon, and good evening. I'm Chloe from RWD Life Science. Thank you for joining our webinar. The RWD Academy is a platform for researchers around the world to share their research achievements. In recent years, the application of techniques such as optogenetics, chemogenetics, fiber photometry, and true photocausal imaging to regulate and detect neural activity in the nervous system of awake, fluid-moving animals has become an important technique for studying memory, emotion, and behavior, neural circuits, and neurological diseases. It's our honor to have Dr. Oliver Barnstedt and Dr. Petzold as the webinar lecturer. Today, they will bring us an academic sharing about illuminating the neural circuits of learned and innate behavior guided by emotion and memory. Here is the schedule for the webinar. During the presentation, you are welcome to submit your questions in the question box. Oliver and Anne will choose some questions to answer in the Q&A part. Especially if your questions are selected to answer, we'll give away our fiber optic candles trial as a gift after the webinar. So don't hesitate to participate and ask questions. We look forward to your joining in the discussion. First, we will have Oliver present his research. Oliver is a neuroscientist and focus on figuring out of the neural circuit mechanisms behind memory, motivation, and behavioral, including the hippocampal striatal pathways and their roles in learning, memory, retrieval, and behavioral flexibility. He combines advanced methodologies of the genetics, in vivo causal imaging, and vitro reactive behavioral paradigms. He leads the research group multiscale circuit analysis at the European Neuroscience Institute Göttingen to link theory mechanisms with complex behavioral. Today, he will present his research on the topic illuminating memories using lighting to dissect hippocampal memory pathways, which focus on exploiting how medial septum VTA activation biases mouse toward exploratory behavior across behavioral states, designing hippocampal accumbens of circuits for spatial reward learning in head-face mouse using in vivo optogenetics and two-photo causal imaging. Now let's get started. Hello, Oliver, can you hear me? Please say hi to everyone and start your sharing.
Hey everyone, I hope you guys can also hear me. Thanks so much, Chloe, for the kind introduction. I will try and get the screen share started. Should be able to see my screen now.
Yeah,
Yes,
works.
can you see everything? Perfect.
Yes.
All right. So thanks again for the kind introduction. Good afternoon, everyone. I would like today to talk about a lab work from my previous lab that was located in Magdeburg and Bonn. Since end of last year, I'm leading the research group Multiscale Circuit Analysis, where we are interested in the neural circuit mechanisms that are linking memory and behavior. So since last year, we're now located in the beautiful town of Göttingen. And if you come by, next month to Göttingen, you might be able to see not far from the main train station, this beautiful view here, which is the Göttingen Christmas market, which is about to start very soon. And if you're to one of these Christmas markets for the very first time, one of the first behaviors you might be engaging in would be to explore this. So you would roam around and see the different booths, maybe try some German sausage or some mulled wine that you see in the booths, or marvel at the beautiful artisan work that people are exposing there. And then while you're roaming around and you're exploring this Christmas market, you might be able to find one of these booths here. This is a delicacy that you normally only find on these Christmas markets. It's called Flammlachs, which is flame-grilled salmon. And if you're like me and you really love this, you might actually remember that location and think that this was really quite a fantastic experience, so that the next day you come to the Christmas market, you might be able to find your way specifically to that specific booth after you've learned that location. Now, if we want to understand how the brain is managing these different behavioral strategies or so, it really helps that not only can our brains perform such exploration and such spatial memory retrieval, but also the rodent brain can. And of course, the rodents wouldn't normally go to the Christmas markets. I hope there are not too many mice around at the Christmas markets, but it's really a general behavioral strategy that under a context of higher uncertainty, mice or also other animals would engage in exploratory behaviors. And once that uncertainty has been narrowed down and the mice know where to find a certain reward, they might actually exploit that and engage in spatial memory retrieval. So we're really interested in the brain's neural circuit mechanisms that are implementing these two behaviors. And I want to spend today to really talk about these two different behavioral strategies and how they're implemented in the neural circuits. And one good starting point for this may be looking at the hippocampus. So most of you are probably familiar with the hippocampus, and it has you can see it here in 3D in the mouse brain with the different subfields. And it has a long history that's been known to be involved in spatial navigation behaviors, but also in spatial memory behaviors since at least the 1970s. So perhaps this is a good starting point to understand and try and figure out how exploration behaviors, so understanding and learning about a spatial environment, and then exploiting such a memory and actually engaging in spatial memory retrieval might be implemented in neural circuits. So, and one important input to the hippocampus, one important modulatory input that has been known and described for quite some time is the medial septum and diagonal band of Broca. And there was really a landmark study from my previous lab and colleagues in Bonn, where in 2015 they showed that if you optogenetically stimulate specifically the glutamatergic neurons in this medial septum and diagonal band of Broca, you can actually initiate locomotion behavior, and you can even trigger and modulate the velocity that the animals would be running on this virtual reality environment on a floating ball. So this seemed to suggest that the medial septum is really important for this locomotion initiation. And you might be thinking, well, then it's perhaps, sending this information over to the hippocampus. But what we found, and what my previous lab also found, is that this pathway to the hippocampus does not actually seem to be driving this locomotion initiation, but seems to be more important for theta oscillations and driving hippocampal theta. That is also important for spatial navigation, but not actually driving such locomotion initiation. So well, we can see that there's these three different subpopulations, glutamatergic, GABAergic, and cholinergic neurons, all of which are important for theta oscillations. This specific role of the glutamatergic neurons in locomotion initiation, which pathways this is actually implemented in, has remained unknown. And it was really then down to a star PhD student in Stefan Remy's lab in Bonn, and a colleague of mine, Petra Mocellin, who has marveled and who has delved into the circuits that are guiding this MSDB information and that are actually mediating this locomotion initiation behavior. And what Petra did, amongst other things, was to inject anterograde transsynaptic tracing virus, in this case AV1-CRE, into the MSDB, and then identified that there's strong axons, and in this case here with synaptic tracing, even neural cell bodies that were clearly present in the VTA. So the ventral tegmental area, of course, is a brain region that's very well known and renowned for its role in reward and motivational states. And if you zoom in here, you can really see that these projections here from the MSDB, from the glutamatergic MSDB population, they're really spread widely throughout the VTA, and they're targeting both the dopaminergic neurons, now TH positive, as well as TH negative neurons. And at that point, it has already been known that the VTA might have a role in locomotion behaviors, because VTA neurons have been found to be multiplexing information about speed, acceleration, and head direction, and that VTA also increases its activity during locomotion, and that the electrical stimulation of VTA seems to be sufficient to elicit locomotion. So it seems to be the case that the VTA might actually play a role with this locomotion initiation, but is it actually the projections from the MSDB to VTA that are driving such behaviors? And the first thing that Petra did to look at this on a functional level was to use fiber photometry. So in this case here, she used a VGLUT2-CRE mouse line to target specifically the glutamatergic population of the MSDB, and injected flex GCAM6 virus into the medial septum, and then implanted optic light fibers above the VTA to measure the axonal calcium activity. So here you see a picture of the injection site, and the mice that we had were head fixed, so we could really have a fine look over the velocity of the animal. And what you could see here in this example is that whenever the mouse here, you see the speed information, whenever the mouse is actually starting another locomotion bout, there is a mild increase in the activity of these axons within the VTA coming from the MSDB, glutamatergic neurons. And if we look at this more systematically across animals, you can really see that whenever the mouse is engaging in running behavior, this calcium activity is increased, and there's even quite a linear relationship, so that the faster the animal moves, the higher also the calcium signal. So this suggested that these axonal terminals within the VTA coming from MSDB might have at least a correlation to the running behavior of the mice. But correlation does not imply causation, of course. Is there also a causal role for this projection? In order to figure this out, we used a very similar strategy, but only instead of flex GCAM now, she injected chenrodopsin, again driven by VGLUT2-CRE, and installed optic light fibers within the VTA to stimulate specifically the projections. And here's a sample video where you see, so this is the behavior without the light on. And then when you turn on the light here, you can see that the mouse on the treadmill steadily engages in walking behavior. And you can also see to some degree, hopefully, that if you stimulate at a faster rate, so in this case 12 Hertz optogenetic stimulation, the animals also seem to be running fast. And if we quantify this, you can actually see that if we stimulate at 3 Hertz, we don't really see an increase in the mean speed. However, if we stimulate at 6, 9, 12 Hertz, you can see that we can actually increase the activity levels, the walking levels of these mice. But is that only, does it only hold true for head-fixed mice? So we also did the same paradigm in freely moving mice. And what you can see here is that, again, if we start stimulating optogenetically this pathway, the mice actually are running more. And interestingly, we also had a look if the reverse also holds true. So if we actually silence this projection, if we get a decrease, and indeed that's what we see a mild decrease in the running speeds in freely moving mice, if we now start inhibiting optogenetically this pathway from MSDB to VTA. So at this point, it really seemed like there was this bidirectional control of movement speed coming from the glutamatergic population of MSDB over to the VTA. But one point that we were engaged in a lot of discussion is, well, when the animals are starting to walk, why are they actually walking? Why would anyone go somewhere if not for a particular reason? So what's the reason? What's the motivation for these mice to actually engage in this walking behavior? And in order to figure this out, we developed some cool computational tools, and we call this one here MouseFlow. And we have a GitHub page, so please feel free to check this out. And what we can do with MouseFlow is to use optical flow as a measure of the movement activity within specific facial regions. And here you see a head-fixed mouse with a close-up view of the face of this animal. And here in false colors, you can see the intensity of movement as tracked by optical flow. And here are our regions of interest around the nose and the whiskers. And what this shows us, outside from and apart from any marker-based approaches, we can really determine with very high accuracy if there's any movement happening on the face here in terms of whisking and in terms of sniffing. And what I hope you can see here is that the moment that you start stimulating, the first thing that is not happening is that the animal would be running or moving, but it's actually starting to move its nose, and it's starting to move the whisker pad. So we see that the animal is, first of all, the moment we start optogenetic stimulation, engaging in sniffing and whisking behavior. And then here also on aggregate, you can see only then later on it is starting to move. And we also quantified this across different stimulation intensities, along different stimulation frequencies. I mean, here in channel dropsin group, here in the control group. And you can see that the magnitude of this optical flow, so the intensity of movement, is enhanced, is getting stronger. It's increasing for the nose here in red, and for the whisker pad here in green, even before the onset of locomotion. Okay, so this was one hint that maybe whisking and sniffing behaviors indicate some kind of exploratory behavior. But of course, there's more to exploration than just sniffing and whisking for mice. So we also looked at our freely moving setup and saw what freely moving mice were doing more closely. And for this, a colleague of mine, Kevin Luxem, developed this amazing toolbox VAME, variational animal motion embedding, where you can, in an unsupervised fashion, see what kind of freely moving behaviors the animals are engaging in. And if we do this, perhaps unsurprisingly, we can see that these sort of behavioral motives here that are implicated in running, or that are part of a running behavioral community, they are upregulated whenever the mouse is stimulated here in blue. But interestingly, it is not only running, but it is also rearing, for example. And you can see here this motif here that is part of the rearing activity is highly significantly increased whenever we stimulate this pathway. So overall, taken together, this suggests that this pathway from MSDB over to VTA, from specifically the glutamatergic subpopulation, seems to promote exploratory behaviors in way of walking, in ways of whisking and sniffing, and also in terms of rearing. So all of these behaviors together seem to suggest that there's really some kind of exploratory behavioral drive that we can specifically causally implement using optogenetics here. But of course, as exploration is just one part of the strategy, because we also just heard before that once we have identified a location that is offering a reward such as food or so, we actually want to engage in specific behaviors in order to get that reward. And we are wondering about the specific pathways that might be mediating this. And of course, if we say and if we know that the hippocampus plays an important role in spatial memory, then somehow this spatial memory needs to be projected onto other brain regions that are then tasked with implementing such exploitation behaviors. And one key region that we identified based on literature was the nucleus accumbens. So we know already for many years that the dorsal hippocampus has direct projections, especially coming from the hippocampal subregion CA1 and prosubiculum over to the nucleus accumbens, as you can appreciate here from the Allen Connectivity Atlas. So we know that there is this anatomical projection. And from studies from the 1990s, we also know that this nucleus accumbens region, part of the basal ganglia, is required, for example, for the Morris water maze, so spatial memory task. And more recently, in 2019, from David Dupre's group, we also learned that for condition place preference, which is a kind of spatial memory task, this connection from dorsal CA1 to nucleus accumbens seems to play an important role. So the question that we really wanted to answer at this point was, what role do these hippocampus accumbens projections play for the expression of spatial reward memories? And in order to do that, again, we turned to head-fixed mice that were running on a Q treadmill belt. So you can see here, and in this case now we trained the mice to lick at a specific location on this belt in order to obtain a milk reward. So in this case here, you see there was a water droplet that the mouse obtained when it was licking in the correct location. And in this case here, the mouse is habituated and it's freely engaged in this task, but it does not really know where to lick yet. However, if you look at day 5 now of the same mouse, you can see that it's running at very high speeds, identifies the correct reward location. So in this case here, this would be the booth with the flame-grimmed cell, and engage in appetitive action that you see here. So now you can see this over 5 training days or so, the number of rewarded laps increases. And if you look at the lick activity here on day 1 and compare that to day 5, you can see that the mouse is engaging more and more in anticipatory lick activity, which indicates that the mouse knows that where the actual reward location is and engages in the appropriate behavior. Now, in order to understand if the dorsal hippocampus to nucleus accumbens projection actually plays a relevant role for this behavior, we again turn to optogenetics. In this case here, we're using a retro AAV that we injected into the nucleus accumbens that is expressing ARCH-T. It travels upstream along the axons. And in this case, we implanted optic light fibers above the dorsal subiculum, dorsal CA1 region, as you can hopefully appreciate here from our histology images. And the task that we did was we, first of all, trained our mice for 4 days along this Q treadmill, where the mouse would receive a reward at the end of this belt, this specific location. And on day 5, we would then optogenetically stimulate only when the mouse was reaching this final stretch toward the reward zone. And the first thing to note is that the relative appetitive licking did not increase over the course of 5 days or decrease or change in any way. And also the overall lick rate did not change in any way if we compare our ARCH-T mice to the control mice when we optogenetically stimulate here. However, what we do see is that if we now zoom in and look specifically at the amount of appetitive licking that the animals are showing in anticipation of the reward zone, you can see that if we now optogenetically stimulate just in front of the reward zone here, we can actually reduce the appetitive licking that is occurring just in front of the reward zone. And if we quantify this here, you can see the significant reduction in the animals expressing ARCH-T compared to controls. So this suggests that in order to retrieve the memory where exactly to engage in appetitive licking ahead of the reward zone, the mice would actually require this, this pathway from dorsal hippocampus to nucleus accumbens. So this is important for the spatial memory expression, but is it also important for the learning? For this, on day 6 to 8, we flipped the reward zone, and now the mice had to learn that 180 degrees on the other side of the belt, that there would now be a reward to be gotten. And if we look at this here in gray, you see the control animals over the course of another 3 days, they learn this quite well, and they come up to about 65% of licking that occurs within this new reward zone. However, the mice that we continuously ahead of this new reward zone optogenetically inhibit this pathway, we see a flat curve. So basically the animals cannot learn this spatial reward location. Okay, so this suggests that this pathway may be necessary, but we're also wondering, well, is it maybe necessary? Is it maybe sufficient? You know, what happens if we actually stimulate this pathway? And for this, we used optogenetics and injected AAV-driven channelrhodopsin into the dorsal CA1, dorsal subiculum region, and implanted an optic light fiber into the nucleus accumbens, as you can see here, bilaterally. And then we started stimulating while the mice are again walking on the treadmill. And I hope what you can appreciate here in this video is that the moment that we start the stimulation, as indicated here in blue or here in the bottom, at the blue trace, we can actually stimulate and bias the mouse towards engaging in lick behavior. So we can get a significant increase in the mouth motion. You see this in this 2 animals, one experimental animal, and here's the control animal. The moment that we start the optogenetic stimulation, the mouse is starting to move its mouth. And we see this also across animals here, if you look at the mouth motion curve, and also if you look at the single data points here across mice. So this suggests that this pathway is not only necessary, but to some degree it may even be sufficient in order to engage in this appetitive behaviors. But next, of course, we wanted to know, well, if this pathway is actually causing the expression of such spatial memories, what information does the dorsal hippocampus actually transmit over to the nucleus accumbens to make the nucleus accumbens engage in such action? And our hypothesis was basically twofold. Well, on the one hand, everyone knows, of course, that in the hippocampus there are place cells. So these are cells that are becoming, that are strongly active at specific locations in space. Once the mouse has learned an environment, each one of these cells here would be active at a specific part of the location. But of course, we know that the hippocampus is doing much more than just spatial information. For example, most recently in 2018, Gautier and Tang also showed that besides the spatial information, there's also a lot of reward information. So place cells that seem to be coding specifically for these specific locations where a reward has to be gotten. So and we thought that perhaps this kind of reward information might be important for a hippocampus and to transmit to the nucleus accumbens. But how can we actually identify the coding properties of specific projection neurons from the hippocampus to the nucleus accumbens? And how can we compare this to a general population? And for this, we used a dual color calcium imaging approach where we injected a retro-driven Cre virus into the nucleus accumbens and then injected DIO M-cherry, so Cre-dependent M-cherry into the hippocampus. And then we implanted a 3 millimeter cannula window over subiculum and dorsal CA1 region. And you can see what this looks like here with the implant. And when you then do this in Thyr1 GCAM6S mice that are paneuronally transgenically expressing GCAM6S, you can see here in green and here in red the retrogradely projected cherry. You can see that we both have the calcium indicator expressed paneuronally, and we also have the specific projection information of those neurons projecting to the nucleus accumbens. And what this allows us then in a freely, in a head-fixed moving mouse is to identify the coding properties of all of these hippocampal neurons while also retaining the information on which of these neurons is actually projecting to the nucleus accumbens and which one is not. And if we do this in one sample experiment here that I would like to showcase, you can see that we can image from hundreds to up to 1,000 or so individual neurons. And these here, for example, would be specifically neurons that are only expressing the calcium indicator. And down here in red, these would be individual calcium traces of those neurons that are also expressing M-cherry and that are projecting to the nucleus accumbens. And if you look at the behavior on top here, there's a lot you can see already from this plot. But the main takeaway I want you to look at is that you can see the sort of sequential activity here of the individual neurons as the mouse is progressing along its track and as it is repeating lap by lap through this virtual reality environment. And you can at the same time now see within the same field of view what is happening with the neurons that are specifically projecting to the nucleus accumbens. And you can see that there's actually a fairly similar pattern. And if we zoom in here, you can also see that this is a very strongly spatially tuned neuron here that is mostly active at this part of the belt. And here, this would be a neuron that would be more active at this part of the belt. And I hope what you can appreciate is that if these neurons that are projecting to the nucleus accumbens were mostly active only at the reward zone, we would see the activity mostly here, but it really seems to be spread across the belt. But what was more surprising to us was that perhaps it's not so much the reward information that seems to play an important role for this accumbens projecting population, but what we could actually find is that whenever the mouse is engaging in appetitive licking as it is approaching the reward zone, every time that we see this anticipatory licking, we see, if you look at the average calcium activity, that this accumbens projecting specific subpopulation shows an increase in calcium activity. And we quantified this also again across our neurons, and you can see the significant increase in the average calcium activity. And also if we zoom in on a single cell level, and identify specific neurons that are statistically significantly modulated by this appetitive licking, you can see the strong increase in that there's a significantly larger part of the hippocampal population that are appetitive lick modulated in the accumbens projecting population compared to the rest of the population. So now I've talked about place modulation and I've talked about lick modulation. There's also speed information that is encoded in these neurons. So in order to tease apart like which kind of coding properties these neurons really have, we built a generalized linear model, and we found that indeed the nucleus accumbens projecting subpopulation actually shows more coding or more neurons that are positively modulated, that are coding for position, velocity, and licking. And importantly, they also encode more of these conjunctions. So different parts of these features, position, velocity, for example, encoded in one and the same neuron. So we wondered, Well, and here's basically a pie chart of this, where you can hopefully appreciate that there's really a lot of this conjunctive coding. So we wonder, like, what's the use of this conjunctive coding? Because current theories suggest that this conjunctive coding actually helps output structures to decode task-relevant information. And if we now build a linear decoder and try and identify the reward zone, as you can see here, if we take the conjunctive neurons and the population code from these conjunctive neurons, we are actually better at predicting, at decoding this specific reward zone. So we think that really there's a specific projection-specific conjunctive code transmitted over to the nucleus accumbens to help in the decoding of this task-relevant information. And with this, I would like to summarize the second part, where we used dual color to photon calcium imaging of hippocampal neurons and the projections neurons to the nucleus accumbens. And we could identify within the same field of view their coding properties. We found that there's more spatial tuning in the accumbens projecting population. There's more speed tuning there. We also see that there's more appetitive licking, and we can also specifically activate this appetitive licking optogenetically. There's enhanced conjunctive coding specifically in the subpopulation of accumbens projecting hippocampal neurons. And this suggests that there's a strongly conjunctive hippocampus accumbens code that drives spatial reward memory-based appetitive behaviors. And with that, I would like to thank the people that have been involved in those two stories here. Mainly my previous postdoc supervisor, Stefan Remy, but also my close colleague, Petra Mucellin, who's now in Berkeley, and many other colleagues at the Leibniz Institute and at the DZNE in Bonn. And like I said, I'm very happy that now, since end of last year, I have my own research group at the European Neuroscience Institute in Göttingen. We are blessed with a lot of great funding and fantastic people that I can't, emphasize strongly enough. And I'm also really happy to say that based on the funding that you see here, especially ERC starting grant from next year, we have multiple positions open for 2026. So please do reach out if you're interested in the neural circuit mechanisms that are linking memory and behavior. And I think we have a Q&A later on, and I'm happy to answer your questions that you have there. Thank you very much.
Thanks for Oliver's wonderful sharing. We'll set aside a few minutes for you to write down your questions in the Q&A section. Now let's have a brief introduction to the RWD Lab Science. RWD is the world's leading manufacturer of the lab science lab instruments and medical devices, specializing in the fields of surgery and nursing, neuroscience research, animal behavior research, pathological analysis, and animal health. Neuroscience research requires the good experimental instrument to help everyone with their experiments. RWD provides neural signal research solutions, including fiber photometry, miniscope, optogenetic system, stereotaxy instrument, and a nesting cell machine. Mini-FM series miniscope can be worn on the brain region of free-moving animals to record the activity of its brain neurons at a high resolution in real time. It helps researchers to obtain high-quality data for the basic research of neuroscience such as emotion, memory, behavior, mental illness, and so on for the needs of the brain science research. Mini-FM series miniscope has the following advantage: timing accuracy, low optic area, high spatial resolution, and exact focus. We provide single-color miniscope and dual-color miniscope, and welcome you to contact us for trial. Now it's time for the Q&A section. You are welcome to ask questions in the Q&A section. Oliver will choose the questions to answer for about 10 minutes. And the questioners will also have a chance to get a fiber optic cannulas as a gift. Now let's give the time completely to Oliver.
Okay. Now I see 3 open questions right now. Maybe I'll start with the question by Elsa Wolfrock. And she's asking about validating the glutamatergic MSDB VTA monosynaptic connections and tracing and our optogenetics techniques. And she's wondering how we could exclude non-specific projections. And I think that's really an excellent question. Well, what I can say is that we took very great care, and so this was basically mostly done by Petra. So Petra took really great care in placing the optogenetics, the optic light fibers, just on top of the VTA. And we did a post-hoc analysis, of course, of the histological sections to make sure that they were placed correctly. We also looked at, if the projections are really specifically ending within the VTA and not, for example, substantia nigra also. And for that, we really went to very fine resolution. So if you look specifically into the supplementary of our paper, we did a very diligent job, if I may say so. And in terms of optogenetics, I think in general, I think that also pertains to a different question. Of course, if you're stimulating at cell bodies, even if you're stimulating optogenetically at axons, there's always the danger that you're basically activating an entire neuron and not just the specific projection that you're interested in. So what we did in terms of the MSDB VTA paper, we also infused lidocaine specifically into the MSDB while optogenetically stimulating these axons. So that basically means that if there were any antidromic activation of MSDB neurons that are then projecting elsewhere into the brain, with the lidocaine, we're basically silencing those neurons, the cell bodies themselves in the MSDB, while only activating these axons. So, and I think that's probably the best that we can do at this point, and that should be fairly thorough. So thank you for that question. There's more questions now, they're coming. Okay, there's a question from Carmen Harry, who's saying that the MSDB, VTA, and hippocampal accumbens pathways both shape exploration and reward learning. That's true. So, two different pathways for two different behaviors. If I think that targeting these circuits could have relevance for disorders, for example, depression or addiction, I would have to go out on a limb here. But basically, I think if you can understand more about general motivational pathways, so in this case, the nucleus accumbens, but also the VTA, of course, play very important roles for motivating us to engage in different behaviors. I think if we have a better understanding in that sense, of course, then this would really certainly help. Specifically when it comes, for example, to symptoms of depression. And when it comes to the nucleus accumbens, of course, it also plays a very important role for addiction. And so, which is kind of a form of maladaptive reward memory. Okay, there's a question from Nicola Bomba, who's wondering about the what makes the glutamatergic population in the MSDB so uniquely suited to control locomotion compared with the other MSDB cell types. The quick question is, I don't know, to be honest. I don't know why nature decided to make this specific pathway to the VTA glutamatergic, but of course glutamate is one of the main excitatory neurotransmitters, so that there's actually an activating role and not just a modulatory role in this sense. When it comes to the MSDB cell types, I might actually also point out that we have a really great preprint on bioRxiv right now that has also delved more deeply into the specific MSDB cell types, because the way I presented it as if there were only three different subtypes is apparently not quite true. So if you look more closely, at the molecular architecture of these MSDB cells, you will find that there's actually much more heterogeneity here, which we have not really covered experimentally at this point, but which in the future might actually yield some interesting findings. So we have more questions here. Mm-hmm. Yes, so there's Siad Mois Jawed, who is interested in the hippocampus accumbens circuits, and he's wondering how stable the coding is across days when the reward location changes. If that's a consistent memories or whenever the mouse is learning in the reward location, if this code also changes. This is something we haven't looked at, but which we want to look at. So, you know, if someone wants to join my lab and work on this question, I'd be very happy to consider that application. And there is a question by Caitlin Gillespie. What motivated you specifically to focus on this circuit? Well, did not, here. This circuit. I mean, there were two circuits. Have you also considered looking at the role of nucleus reunions in this behavior? So I guess that Caitlin talks about the nucleus accumbens. And the great motivation for that was mainly a 2018 paper from Nelson Sprusten's lab that has showed that the part of the subiculum has really strong projections to the nucleus accumbens, and then aligned with Stephanie Trusch's data suggesting that dorsal CA1 projections to the nucleus accumbens play an important role for spatial reward memory. This really prompted us to look more closely at what these neurons are actually doing, if they exist this pathway, and it seems to play an important role for spatial reward memory. That was the main motivation. The role of the nucleus reunions we have not looked at. As far as I understand, it is one of the main thalamic relays between hippocampus and frontal cortex regions. And it might play an important role there. It definitely plays an important role in other kinds of spatial memory, but it plays an important role for spatial reward memory. We haven't tested, and I would be hard pressed to find which papers would have looked into that. But it's well possible, and it might serve as a relay to prefrontal cortex. Thanks, Caitlin. So what else do we have? More questions. Is there an ethological? So we have a question by Devanshu Nandi. Two questions. Okay, you could skip this second. Let's see. Okay, is there an ethological way to distinguish goal-directed exploration from habitual exploratory behavior in mice, given their natural tendency to be highly exploratory? Well, first of all, I would disagree. I would say that mice are not naturally particularly exploratory or have a high exploratory tendency. I think working with rats would change that fact, but naturally mice are actually fairly anxious and would only explore if they feel very confident and not anxious. To distinguish goal-directed exploration, I mean, what we have looked at, for example, would be to see if, for example, we provide food stimuli or social stimuli for the animals, and to see if they actually start going towards those locations more, which would, could represent a kind of goal. And so in, I think, the supplementary of our paper, and the Neuron 2024 paper, we did not see any of these goal-directed behaviors. Of course, that we can't look inside the animal's mind. Maybe there is something that it wants to find and not just explore the environment, but as far as we could tell, we did not really see that they were seeking anything in particular. Okay, I can also answer the second question. Are you concerned that optogenetic experiments targeting only a single brain region might not reveal the full picture due to potential downstream effects on connected areas. Well, I mean, I don't think with any of these studies we're really getting the full picture, right? We're only finding little puzzle pieces, and by looking at our experiments, those of colleagues, integrating all of the data, hopefully we can come to a full picture. Downstream effects on connected areas, obviously, I would expect there to be downstream regions that are connected to this and that are mediating this. And we have some speculation as to which regions this might entail. So yes, I don't think that's a problem. I think that's part of the work. Thank you. Okay, and there's another question. So Siham is asking if the optofiber is not impairing the behavior that I'm studying because the fiber is thick. Okay, I think that's a fair question. So of course, so Siham is also suggesting that if we were to do chemogenetics, for example, then with chemogenetics we would not do any damage to the brain. So in general, what we implant is 200 microns of optic light fibers. So the damage, if you look histologically into the brain, should be fairly minimal at that point. We're also taking great care to really lower these implants very slowly. Of course, chemogenetics is an important tool, and we're also employing that regularly. There are several problems with chemogenetics. For example, if we want to specifically inhibit this pathway while the animals are navigating towards a certain reward location, this would have a temporal resolution, or now we're having a temporal resolution on the millisecond basis, whereas if we work with chemogenetics, we would only have a temporal resolution of several hours. So obviously, this kind of very specific inhibition while the animal is doing something very specifically closed loop, that's not possible with chemogenetics. The other problem with chemogenetics would be that it's very hard to target specific circuits without actually implanting any fibers. So, that is, again, not an option. If you don't implant fibers and you give CNO, for example, systemically, then you basically only perturb, so activate or inhibit several neurons. So even if you use, for example, retro approaches, retro-Cree and Cree-driven approaches to target a specific circuit, they are still collaterals or so that you would not be able to not target. Yeah, that would be included.
Okay, Oliver, due to time limitations, we can end the Q&A section ends here. Okay, we will next for the next part. Thank you to Oliver for the excellent report and the answers. And please take a break. Now let's welcome Anne to give the next presentation. Anne is a neuroscientist at the European Neuroscience Institute in Göttingen, and she leads the brain-body interaction group. Her work focuses on the neural circuits that let behavior adapt to changing need states. To study this, she used techniques like single-cell causal imaging in fluid-moving animals, optogenetics, chemogenetics, and molecular tools. Today, she will present how hyperthalamic circuits overcome anxiety. She will focus on the role of hyperthalamic prefrontal circuits for the regulation of the anxiety behavior under normal conditions in the neuropsychiatric diseases models. Especially, we will hear how optogenetics and causal imaging experiments in fluid-moving mouse demonstrate how leptin-sensitive neuron in the lateral hypothalamic counteract axial genetics prefrontal input and enable healthy behavioral under anxiety. Let's begin. Hi, and can you hear me? Please greet everyone and start your presentation.
Hi, Chloe. Yes, I could hear you well. Share presentation. Yes, thanks, Chloe, for this nice introduction and for the invitation to present at this webinar. Oh, great. I hope everyone can see my screen and hear me well.
Yes,
So
we
then
can
in
see
the
your screen.
interest, great. And in the interest of time, let's dive right in. So I am very much interested in the neural circuits that govern behavior. And when we think of behavior and behavioral regulation, especially in regards to human behavior, then we often tend to put behavior in a fairly cognitive context. Think of decision making, learning and memory, navigation, etc. We just heard this wonderful presentation by Ali, for example, like how higher order processes like learning, spatial memory shape exploration behavior. However, I'm here to remind you that even in the sapient human, most of our day-to-day behavior is made up of fundamental innate behaviors that we share with all other mammals and that we do largely on autopilot and that are guided by bodily signals that tell us what the body needs. Now, classical examples of these fundamental innate behaviors are eating and drinking, for example, and many social and sexual behaviors are also innate. Of course, there is a learning component in innate behaviors as well. Of course, they are shaped by experience and the preferences that we form in the course of a lifetime. But they are also very much shaped by our needs, the physiological needs like hunger and thirst, for example, and also, of course, emotional needs as well. Now, a brain region that I have worked on a lot that is really deeply involved in the regulation of innate behaviors is the lateral hypothalamus. We know how important the lateral hypothalamus is for generating the motivation to change or adapt innate behaviors according to internal drives such as hunger and thirst from really rodent studies that were done really decades ago. Really fundamental studies that I think every neuroscientist should know. Back in the day, people often implanted electrodes to lesion parts of the brain. So you apply very strong currents through wires here. You can do this to lesion the lateral hypothalamus, for example. If you do this, then animals will stop to drink and stop to eat. They will generally otherwise be fine, but of course, eventually die of starvation and dehydration. Now, conversely, you can also use such wires to just gently stimulate electrically the lateral hypothalamus, for example. So think of these experiments as like forerunners of the genetic experiments that we do nowadays. If you electrically stimulate the lateral hypothalamus and you let the animal self-stimulate by pressing a lever, then animals find this really rewarding. They will even cross food shock grids to receive stimulation of the lateral hypothalamus. And it was really fundamental studies such as these that showed how deeply the lateral hypothalamus is involved in generating the motivation to do anything, especially eating and drinking. Now, it's of course extremely fundamental for a healthy life that we are able to balance different motivational needs. And it's really a hallmark of many neuropsychiatric disorders that patients have great difficulty in balancing different needs, different motivations, especially if we're thinking of mood disorders such as depression or eating disorders such as anorexia nervosa, where really negative emotions, negative emotional drives, for example, oftentimes get in the way of performing really healthy, important behaviors such as normal eating, for example. Yeah. So an important job of the lateral hypothalamus is to translate bodily signals that indicate the needs of the body and to resolve conflicts among conflicting motivational drives. Okay. So generally, of course, the body has a host of peripheral signals that indicate the physiological status. These arrive in the hypothalamus by way of the ventricular system, and the lateral hypothalamus harbors a range of really very different neuronal populations that are able to sense these peripheral signals and process them, integrate them, and then talk to other brain centers in order to change behavior accordingly. Okay, just to illustrate this in a more concrete way, one signal I have worked on quite a lot in the last years is leptin. This is a hormone. Your fat cells produce it when you eat. Your fat cells produce leptin and secrete it into the bloodstream. Also, the more fat cells, the more your fat cells are filled, the more fat mass you have, the higher is your leptin level as well. Leptin is secreted into the bloodstream and then filtered into the cerebral spinal fluid and then reaches the brain via the ventricular system. And in the hypothalamic areas, especially also in other parts of the brain, we have neurons that express a receptor for leptin. We call them Leper cells or leptin receptor cells for short. These are neurons that are located mostly in the medial hypothalamus, such as the arcuate nucleus, very important brain region for the regulation of eating behavior, and also the lateral hypothalamus. And I'm going to talk a lot about the role of leptin receptor neurons in the lateral hypothalamus in the regulation of behavior. Now, we know that these leptin receptor neurons in the hypothalamic areas are very, very important for the regulation of eating behavior because really fundamental studies from a few decades ago infused leptin directly into different hypothalamic areas, including in the lateral hypothalamus, and observed that this manipulation would decrease eating behavior. And it's from such studies that this idea was formed that leptin is this quote-unquote satiety hormone, although you will see that it has a lot of other functions as well. Okay, from these early electrolytic lesion studies and also the pharmacology studies, these were really fundamental, but they left open many other, many very, very important questions, such as what are the actual neuronal mechanisms that enable these leptin sensitive populations, including those in the lateral hypothalamus, to sense these changes in the energy state, sense changes in leptin levels, for instance, and adapt feeding behavior. Okay, so I'm going to answer some of these questions here. Now, if we want to understand how these leptin-sensitive circuits are involved in the regulation of eating behavior across different need states of the animal, such that when it is hungry and when it is thirsty or when it is sated, for example, then we need to be able to record the activity of such cells, specifically of this particular subpopulation, this leptin receptor neurons, and we need to do this as the animal is freely engaging in spontaneous innate behaviors, including eating behavior according to its natural motivation at this time. And ideally, we want to do this across different states of the animal. So we want to at best follow the same cells. Okay, and this is why for this particular project, I turned to single-cell calcium imaging because it's really a technique that covers all of these experimental requirements. So what we do here is we take a leper-Cree mouse line, and in the lateral hypothalamus, we are injecting a Cree-dependent fluorescent calcium indicator. Here at the time, we're using GCAM6M. And then we implant a green lens above the lateral hypothalamus that bridges the gap between the lateral hypothalamus and the miniscope that we can put on top of the head of the animal. Yeah, so it looks like this. The miniscopes are also small that the animals can wear them, and that's really ideal. So if you think about it, these are essentially fluorescent wide field microscopes, just extremely miniaturized. And that's great because in this way, we can really record the activity of leper cells as the animal is really moving around, starting to eat, starting to drink, and doing, what it spontaneously would do according to its natural motivational state. Now, here in the middle here, you see a completely unchanged, completely raw video of how a miniscope recording could look like. So you can appreciate that we have to go through several pre-processing steps in order to extract calcium transients of individual cells from these videos that we can then relate to behavior. But I think it's all worth it because the data sets that we get out of this technique are really extremely valuable to understand neuronal functions and especially to follow the neuronal dynamics across different states of the animal. The part of the presentation about this particular project, the regulation of food seeking feeding behavior by leptin receptors, leptin sensitive circuits, I want to keep relatively short because it's been published for some time now. Suffices to say that through the calcium imaging recordings, we found that leptin receptor neurons in the lateral hypothalamus, they are largely inhibited by food. And the more food inhibition we observed among LEPR neurons, the more food the animal was able to eat. Okay, so it's suggesting that it's actually a food inhibition of LEPR neurons is conducive to eating, which would be consistent with this idea that leptin is a satiety hormone. And it would conversely suggest that the activation of LEPR neurons is one of the ways in which we induce satiation or a decrease in eating behavior as far as we can observe this in mice. Okay, so to test if this was actually the case, we performed optogenetic experiments. So here we're taking again leper-Cree mice and inject channelrhodopsin into the lateral hypothalamus and then activate these cells through an optic fiber. Okay, and then in this arena here, you see the mouse. And then on the left-hand side, this is where we have the food stimulus. This mouse is fairly hungry, so it will start to eat fairly soon. We test the animals under two conditions, when they are sated and when they are hungry. And of course, they will eat more when they are hungry. But when we activate leper cells, then we preclude this increase in hunger-induced feeding, this homeostatic feeding, okay, showing that leptin receptor neurons in the lateral hypothalamus are maybe one of the populations that are involved in the leptin-mediated suppression of eating or its satiety effects. But I told you that I was most interested actually in situations of conflict when different motivational drives were in conflict of each other. So what I do in these experiments is I give animals access to other rewarding stimuli as well that mice also naturally like to explore, not just food that you see here on the left-hand side, but also water that's here on the right-hand side. On the left-hand side here, we have like small objects. These are paper clips as control objects. And then here on the right-hand side, this is a little cage that contains another mouse. So introducing here a social stimulus as well. And the way we analyze the behavior of the animal now is that we measure how much time do the animals spend in each of these, exploring each of these stimuli, the food, the water, and the other mouse. Generally, if we're looking at the control group, then the animals spend most of their time exploring the other mouse. Yeah, so if you're a mouse, then the most interesting stimulus I could possibly give you is another mouse. But of course, if I make the animals hungry by taking away the food overnight, then I take away priority from social exploration and they will explore the nutritional stimuli more. But in the experimental group where we are activating leper cells, now in the sated condition, they also show preferential social exploration, like the control group, but they retain preference for social exploration, irrespective of being hungry, in the hungry condition. Showing that leper neurons, they relegate feeding, they decrease feeding behavior, but they do this or they in turn benefit or prioritize social interaction. Okay, that was really one of like the first experiment that showed that any leptin sensitive population in the brain is involved in the regulation of social behavior. Now, I want to cut this story quite short because it's been published for some time. If you're generally interested in innate behavior control by hypothalamic circuits, then I encourage you to read the paper. There's a lot more in it. We are also contrasting our results on leptin receptor neurons with experiments in neurotensin neurons, another very important and for feeding behavior, very relevant population in the lateral hypothalamus that we show has different and quite complementary effects to leptin receptor neurons. Something that I should really mention for completeness here at this stage is that there are very strong sex-specific differences in almost any innate behavior that we're looking at. And this is, of course, especially true for eating behavior and social and sexual behavior as well. There are also, and that's known across different mammalian species, including humans, rats, and mice, very different leptin levels in males and females. So females have about 3 times the amount of leptin as the males do. So the difference in absolute leptin levels is much higher than you would expect from the fact that female members of the species usually have higher fat content and different distribution of fat. Now, in a study that we are now wrapping up, we are now investigating really what are the sex-specific differences of leptin-sensitive circuits for the regulation of eating and social-sexual behavior. So I'm just mentioning this now because I think for completeness, we have to keep in mind that there are very important sex-specific differences also in terms of leptin-mediated behavior. Now, something that we left completely out of the picture here so far is that many innate behaviors, of course, they are not just shaped by these physiological needs, hunger and thirst, for example, but of course also by emotional states. Yeah, and especially anxiety behavior, or anxiety is a very, very strong emotion that can prevent, hinder or even prevent other very important innate behaviors, such as eating, for example. Now, there are situations where this is completely adaptive and essential for survival. But of course, there are many mental health disorders where negative emotional states really get in the way of the expression of very important healthy behaviors. We see this a lot in anorexia nervosa, for example. This is, of course, a restrictive eating disorder. It's characterized by patients not eating enough, of course, and many also engage in way too much exercise, which is, of course, completely maladaptive because it exacerbates the caloric deficit that they already have. Now, there's again a very strong sex bias in many neuropsychiatric diseases, including in anorexia nervosa. It's much more prevalent in females, although health outcomes are oftentimes even worse for male patients. Anorexia nervosa is one of those neuropsychiatric disorders with a very high comorbidity with anxiety disorders. And in fact, the idea in the field is that anorexia nervosa patients use this excessive exercise because it's anxiolytic, because it gives them relief from excessive anxiety levels and negative emotions. Now, we got interested in the relationship between leptin and leptin-sensitive circuits and anxiety, including in the context of anorexia nervosa, for 2 main reasons. One is that in anorexia nervosa patients, leptin treatment can actually be beneficial. Now, if you think about it, it's completely paradoxical. It doesn't make any sense. Why would you give a satiety hormone to someone who already doesn't eat enough? The observation is that leptin treatment in anorexia patients can actually give them some relief from negative emotional states. It can be anxiolytic and it has some antidepressive properties. And this is why it can be beneficial. Although the leptin sensitive circuits that could explain such an effect are completely elusive. What we do know is that many anorexia patients suffer from overactivity of the prefrontal cortex, and this seems to be related to excessive activation by what is for them anxiogenic stimuli, like food and eating related imagery, for example. Although again, what's the relationship between prefrontal cortex circuits and leptin sensitive circuits, including in this context, is quite elusive still. Now, the second fact that got us interested in this topic here is that we actually see similar effects of leptin treatment in animal models. So, of course, it's not that easy to model neuropsychiatric disorders in animals. There are always some shortcomings. You cannot model everything. The way we model anorexia nervosa in mice, and works very similar in rats, classically is the activity-based anorexia model. The way this works is that you give the animal access to a running wheel, which they love using voluntarily. They love it, and all is fine. And then after a few days, you take away the food, and you only restore food to the animal for 1 to 2 hours a day. It depends on the specific protocol that you're using. And what you see in response is that the animals will start to run excessively, which is, of course, completely maladaptive. Why would you expend much more calories if you're already in caloric deficit? Because of this combination of symptoms, a caloric deficit leading to excessive exercise, that this model is used to model anorexia nervosa in humans. Now, to show you how this would look like in an experiment is we're looking here at locomotion levels of the animal. This is usually like wheel turns. And then when the animals are fasted here, this is when they show this excessive increase in exercise that then relaxes back to baseline when the animals are released from food restriction. Now, interestingly, these excessive levels of exercise that the animals are showing here seems to be also related to the anxiety levels, and it could be quite similar to what we see in patients as well. Now, when animals in this model are treated with leptin, then this manipulation decreases the levels of excessive exercise. Yeah? So if we take these observations together, it seems that leptin treatment improves or can improve anorexia nervosa symptoms in humans and also in animal models, although there are many important questions that are still completely elusive. One is, for example, what are the neuronal substrates? What are the leptin sensitive circuits that are responsible for such effects? How do they get anxiogenic information? How do they integrate anxiogenic information with nutritional information? And can we harness knowledge about the leptin sensitive circuits that are at the core of this phenomenon for therapeutic purposes? And I will now in the second half of the talk touch upon some of these questions. Now, we are interested especially in the role of leptin receptor neurons in the lateral hypothalamus in the context of anxiety and anorexia nervosa. Because when we more deeply analyze the molecular profile of leptin receptor neurons from transcriptomic data, and we pulled out the leptin receptor positive neurons of the lateral hypothalamus, that's here clusters 3 and 4, we find that these neuronal groups express 2 human anorexia nervosa risk genes at very high levels. And especially for one anorexia nervosa risk gene, EBF1, there are antibodies available, and we have confirmed co-expression histologically. Okay, but what could be the role of leptin receptor neurons in the regulation of anxiety in general and in anorexia nervosa in particular? If you have worked in the hypothalamus field for any amount of time, you will know that it is not classically involved in the regulation or known to be involved in the regulation of anxiety behavior, especially if you're thinking outside of orexin neurons. So I first wanted to know, take a step back and want to know, okay, do leptin receptor neurons in the lateral hypothalamus actually respond to anxiogenic information in any way? And so here we performed single cell calcium imaging experiments in the elevated plasmase. That's the classical anxiety test in mice. And we looked especially how would the leptin receptor population respond when the animals entered the open arm of the elevated plasmase, which is of course the anxiogenic zone, the exposed space. Generally, across the population, leper cells were strongly activated when the animals were entering the open arm. We actually find a quite high proportion of open arm excited neurons that was much higher than the closed arm excited population, although, of course, the animals spend much less time in the open arm. Okay, so does that mean that leper cells are actually involved in the regulation of anxiety behavior? We can test this question optogenetically. So here I recruited a PhD student who was working with me for quite some years now in the Korotkova lab. we were injecting again channelrhodopsin in leper cells of the lateral hypothalamus to activate those cells optogenetically in this case, and then tested the animals in the elevated plasmase. When we activate leper cells in the lateral hypothalamus in the elevated plasmase, animals are willing to spend much more time in the open arms, and they also go to the open arms much more often in the first place. And we had the same observation when we activate leper cells chemogenetically as well. Altogether showing that leper neurons, they do respond to anxiogenic environments and stimuli, and they actually also seem to facilitate the exploration, in this case, of exposed spaces, which is for a mouse, of course, an anxiogenic environment. Okay, we're also wondering, of course, then about the specificity of this phenomenon. Is this specific to leper cells, or would we observe this if we looked at any population of lateral hypothalamic neurons? Now, lateral hypothalamus is the harbor of many, many different neural populations, not all of which I can test in my lifetime. We did test anxiogenic or anxiety-related responses in neurotensin cells, at least, because that's a very relevant other population in the lateral hypothalamus that is known to also be involved in feeding behavior and in a complementary manner. So it seems to be like the closest population to really compare leper cells to. When we record the activity of neurotensin neurons using miniscopes in the elevated plasmase, we do not see that these cells are specifically activated when the animals enter the open arm. And we also do not observe a very high open arm excited population. And when we activate the neurotensin population, in this case, in the open field, which is another classical anxiety test in mice, then the animals do not go much more or less into the center of the open field, or they do not take more or less time to enter the center of the open field in the first place. This was actually done by Hanna van den Munkhoff, who at the time was another PhD student in Tatjana Korotkova's lab and who's now a postdoc there. Now, if we take the neurotensin data together, conclude that this other feeding relevant LH population does not respond to anxiogenic stimuli and does not facilitate their exploration. My conclusion from the sets of experiments on leper and neurotensin cells so far then would be that the observation that leper cells respond to anxiogenic stimuli and seem to be involved in the regulation of anxiety behavior is, as far as this comparison goes, specific to leper cells. Now, how do leper cells sense anxiogenic information? What are the sources of anxiogenic information to the lateral hypothalamus? Now, I have to say, generally, inputs to lateral hypothalamus are not very well described. So we would be left wondering a bit what could be the potential sources of anxiogenic information. But we were very lucky that Zane Andrews' lab had just published a paper at the time where they showed that PFC neurons that project to the lateral hypothalamus increase anxiety behavior in mice. So that gave us first plausible target to look at, the prefrontal cortex. We first wanted to check whether the PFC inputs to the lateral hypothalamus actually convey anxiety-related information. We did this by way of hyperphotometry recordings. This was done by Alisa Bakareva, another PhD student in the Korotkova lab. She found that when she recorded the activity of the inputs of the prefrontal cortex in the lateral hypothalamus with hyperphotometry in the elevated plasmase, that these inputs were activated as the animals were entering the open arm quite systematically across animals. And she also did the optogenetic experiment. She also activated the inputs of the prefrontal cortex into the lateral hypothalamus in the elevated plasmase. And this manipulation actually decreased the time that the animals were willing to spend in the open arm and how often they went into the open arm in the first place. Okay, so altogether showing that indeed prefrontal cortex conveys anxiogenic information to the lateral hypothalamus. Now, the next question would be, how do leper cells actually respond to such input, right? We can test this by combining single cell calcium imaging of leper cells with the optogenetic stimulation of PFC inputs. So what we do here is we are expressing a redshifted opsin, CHR-MINE, in the prefrontal cortex. That is then transported along the axons into the lateral hypothalamus, and we're expressing GCAM6M, the fluorescent indicator, in leper cells of the lateral hypothalamus. Okay, and then we implant the green lens above the lateral hypothalamus, and we provide the light stimulation for the imaging and for the optogenetic stimulation in the same throughout the same lens. In the recording, we are combining periods where we stimulate and periods where we do not stimulate, so we can really assess the responses on the individual level of the individual neuron. Generally, leper cells were inhibited by PFC input, and the more leper cells were inhibited by PFC input, the more anxious was the animal. Yeah, or if I phrase this in other words, leper neurons are generally inhibited by PFC input in a manner that scales with anxiety levels. Yeah, indicating that indeed PFC input provides anxiogenic information to the lateral hypothalamus and increases anxiety behavior by inhibiting leper neurons. Okay, but that would be a hypothesis still. Okay. Now, would this anxiolytic activity of leper cells mean that they could counteract such anxiogenic input in order to enable an animal to engage in adaptive innate behaviors despite the fact that it is in anxiogenic conditions, such as, imagine your mouse, you have to go into an exposed space in order to find food, but of course you're anxious that something bad will happen to you, so to speak. Now, if we put this more scientifically, we can test this question in a particular task. It's called the novelty suppressed feeding task, but it works quite as I described already. So you put the animal in an open field. This is a rectangular arena typically, it can also be round, with quite strong light levels. So animals do not really like to go into the center because they are afraid it's an exposed space, but you put the food in the middle. Yeah, directly in the center, and the mouse is hungry. So it needs to go into the center in order to eat. So you're putting the animals in this direct conflict between feeding drive and anxiety. Okay, now Rebecca then recorded the activity of leper cells under these conditions. Generally, leper cells were activated by this food in this anxiogenic space. And then we lined up the activity that we saw of leper cells to the behavior. And we're looking at two behaviors here specifically shaded in white. This is novelty suppressed feeding that we call it in this scenario. So episodes where the animals go to the food, but they refrain from eating. And then here shaded in yellow, this is what we call, for reasons of simplicity, successful feeding. So the first time that the animals actually go to the food and they start eating under these conditions. And these episodes are preceded by strong activation, endogenous activation of leper cells. Now, the hypothesis would be that this endogenous activation of leper cells would provide enough anxiolytic anxiolysis, so to speak, in order to enable the animal to eat under these anxiety-inducing conditions. And then if this hypothesis were true, then I would expect that if I exogenously activate leper cells in this scenario, then I would improve the ability of the animal to start eating. And we can just test this now chemogenetically. Here, Rebecca teamed up with a bachelor student in the lab, Nele Hugger, and they expressed the chemogenetic actuator HM3DQ in leper cells. And in the control group here, and this is in the gray line, we see that it takes the animals typically quite a long time to start eating under these conditions. But when we're activating leper cells, then animals start eating much earlier. Okay, altogether showing that indeed leper neurons, they do facilitate feeding under such anxiogenic condition. Now, of course, the million-dollar question is now, does this have any bearing on anorexia nervosa, right? Does the sensitivity of leper cells to anxiogenic conditions relate in any way to the risk or to the expression of anorexia nervosa symptoms? Okay. How do we test this? Well, we have already performed the elevated plasmase task with our animals, so we can phenotype them as high or low anxiety animals. And then we can test them, both the high and the low anxiety animals, in the anorexia nervosa model. Yeah, just to remind you, the way this works is that we give the animal access to a running wheel. They will start to run naturally and all is well. In the actual experiment, it would look like this. Then we take away the food and we only give food to the animal for 90 minutes a day in this case, and they will start to run excessively. Okay, and then what we have observed, and I'll show you the data in a moment, that the excessive exercise levels that the animals are showing reflects how anxious they are. Okay, now the question is, does the natural activity of leper cells also reflect the anxiety level of the animals under these conditions in some way? Okay, so luckily we can combine, and this is why the animal here has a mini scope on top of the head. We can combine the anorexia nervosa model here with mini scope imaging and record the activity of leper cells towards food and towards the running wheel and during running. And what we observe is that we can actually predict whether we are recording from a highly anxious or not so anxious animal based on the activity of leper cells in the activity-based anorexia model towards food and towards the running wheel alone. Yeah. So if I phrase this in another way, the leper activity that we record under these conditions, it reflects anxiety in this anorexia nervosa disease model. Now, does this mean that leper cells may have any therapeutic utility? So just to remind you, we already know that, generally speaking, from pharmacological studies, if we inject leptin in the anorexia nervosa disease model in mice, then we improve these excessive exercise levels. Okay? Now, could leper cells in the lateral hypothalamus play any role in this? To answer this particular question, we again turn to chemogenetics and recorded the excessive exercise levels of the animals while we are activating leper cells. We're looking at the control group here in gray. We see, yeah, when they have access to a running wheel and they also have food, they run, but they don't run excessively. Then we take away the food restriction starts, and this is when they increase exercise levels very highly. Okay, and then we give the food again normally, and then locomotion goes back to baseline. Okay, but in the experimental group here, we are activating leper cells. We activate them each day, so we're injecting CNO every day, while the animals still have food. This does not really change, manipulation does not really change baseline locomotion levels. But when we food restrict the animals, when the food restriction starts, then the activation of leper cells precludes excessive exercise levels of the animals. We also related exercise levels of the animals to the anxiety that we had measured in the elevated plasmase before we started with the anorexia nervosa disease model. So typically, and this is what also previous studies have suggested, the higher is the anxiety of the animal, the more excessive is the exercise level that it shows under anorexia nervosa disease model conditions. But when we activate leper cells, then we disrupt this relationship. Yeah, then the anxiety score or the anxiety of the animal does no longer predict the excessive exercise that it will show, and we generally reduce these excessive exercise levels. Yeah, or in other words, the leper activation precluded the excessive exercise that we see in the anorexia nervosa disease model. Now, in order to sum this all up, what I have just shown you, I have talked a lot about a hormone, leptin, that is secreted by fat cells and then secreted into the bloodstream and filtered into the cerebrospinal fluid to reach the hypothalamus, including the lateral hypothalamus, where we have leptin receptor neurons that are sensitive to leptin, they play an important role in the satiety effects of leptin. Their activation reduces food intake, but surprisingly and very interestingly, their activation increases also social behavior as a flip side of the coin. The lateral hypothalamus and especially leptin receptor neurons receive information from the prefrontal cortex that is related to anxiety. Prefrontal cortex input to lateral hypothalamus actually drives anxiety, increases anxiety behavior, and generally the prefrontal cortex input inhibits LAPA neurons, and conversely, the activation of LAPA neurons is able to block such anxiogenic input from the prefrontal cortex. And these anxiolytic properties of LAPA neurons, they can have beneficial effects and improve the ability of an animal to perform adaptive behaviors under anxiogenic conditions. We have tested several scenarios and found that the activation of LAPA cells facilitates the exploration of exposed spaces, for instance. It enables eating under anxiogenic conditions, and it improves symptoms in a preclinical anorexia nervosa disease model. And of course, the hope is that we can take such insights about the role of leptin-mediated behavioral adaptation to eventually inform, of course, clinical studies that may be able to use leptin treatments for the improvement of neuropsychiatric disorders. Now, leptin treatment is already used for the treatment of metabolic disorders, and maybe in the future, eventually, we should at least test whether we can also test it to benefit some neuropsychiatric disorders as well. Okay, and with this, I would like to close. Thank you so much for listening to me for such a long time. I have since opened my own lab in Göttingen, in Germany. We have been very lucky with funding. We have several positions open right now. I encourage you that if you're interested in the neural circuits that govern innate behavior, especially innate behaviors, that you reach out or send students our way, and we're always happy to talk and engage. Now, if you have any further questions, I would be very happy to answer them.
Okay, thanks to Anne for the excellent research. Just mentioned, the RWD can provide neurosignal research solutions, including fiber photometry, miniscope of the genetic system, stereotactic instrument, a net sensor machine. Now let's have a brief introduction to the RWD's fiber photometry system. The RWD's fiber photometry system and miniscope can be used in similar scenarios such as calcium ion and neurotransmitter signal recordings, neurocircuit research, neurological diseases research, and probe development detection. We have multi-channel fiber photometry combined with optogenetics, 4-color multi-channel fiber photometry system. The fiber photometry has the following advantage: 1 choreographs, professional data analysis, high interaction, and strong expansion. RWD's fiber photometry has worked in over 500 labs around the world, and we users from all over the world, such as MIT, Stanford University, Duke University, and so on. Welcome everyone to search our website and have a try. Now is for the Q&A part, and we'll choose some questions from the audience to answer for about 10 minutes. Now let's give the time to Anne.
Thanks, Chloe. I already see a number of very good questions. So let's see. So the first question here is from Jean Dubois. I hope I pronounce your name correctly, who wants to know whether there's a delicate balance between AGRP and leptin that controls the whole feeding behaviors. And of course, Sean, you're right. This is definitely true. There are wonderful studies, including from the Brüning lab at the Max Planck Metabolism Research, for example, but also other very excellent labs that show that you have two populations of neurons in the arcuate nucleus, so that's medial to the lateral hypothalamus. that include AGRP neurons and POMC neurons. They both express also leptin receptors. The activation of these neurons, or the leptin receptors of these neurons, has really antagonizing effects, and it is their, so to speak, their balance, their yin and yang dynamics that are extremely important for the control of feeding behavior. Yeah, so I very much encourage you to read up on arcuate nucleus functions, AGRP neurons, and leptin to learn more about this. Now, the second question is by Bella Taylor, and she wants to know whether miniscope imaging in 3D moving mice is technically challenging. That's true. And whether the experiment can actually affect the behavioral states such as anxiety and could confound, so to speak, our readings because we have this head-mounted device. I think it's a very, very relevant question. Yeah, exactly. So when we compare the anxiety, so the problem we have with animal experimentation, right? We cannot ask a mouse, how anxious are you? The only thing we can do is we can put it in a test that we as humans assume tests very well the anxiety of the animal. And there are classical tests that are used for very good reasons for these purposes. This is especially the elevated plasmase test and the open field test, for example. And we have been using these classical tests, and usually both of them. What we see in our miniscope experiments, that if you're looking across the population of animals, they behave in a very similar way to those animals that do not have the head-mounted device. Yeah, so the data is within the same range, and we also have similar proportions of high and low anxiety animals. So I would argue that generally speaking, the manipulation does not very strongly affect the anxiety behavior of the animals. Although, of course, I have to say that we have to habituate the animals very generally, not to the elevated plasma specifically, but of course to manipulations such as being fixated to put the implant on, for example, right? So you do have to have some measures to make sure, or some procedures to make sure that you decrease anxiety levels, really, generally speaking, to your animal. Yeah, but if we do this, then generally we see relatively little effects on anxiety baseline levels, really, from these manipulations. Yeah, that's also a very good question from Emily Taylor, no? Whether leptin and leptin receptor neurons modulate anxiety directly, or whether they primarily alter behavioral prioritization. Yeah, exactly. This is really at the core of these projects. Well, I think it's both, really, because what people have shown previously, that if you inject leptin in your animals and then you perform classical anxiety tests, such as the elevated plasmase, you see effects in this particular context. Yeah, because you do not give other stimuli under these conditions that the animals can engage with. You would conclude from such experiments, as far as we can, from an animal experiment, that we have direct effects on anxiety. And we also see the same when we are activating leptin receptor neurons optogenetically in the elevated plasmase. We see direct effects on anxiety. If we give more stimuli to the animals and it has the opportunity to perform behavioral prioritization, because there are different stimuli to prioritize, we see effects both from leptin injection and leptin receptor neurons. So if we take all of this together, I would argue that we see both direct effects on anxiety from these manipulations, as well as effects on behavioral prioritization. Yeah, so this is also a very good question from Carmen Harry. They want to know, I'm just reading the question, in anxiogenic environments such as the open arms or the novelty suppressed feeding task, LEPR neurons remain food responsive. What mechanisms enable this resilience under stress? I think that's a wonderful question. And I think this is really one of the next projects that should be done, to actually find out what is there in the PFC to LH, especially the PFC to LEPR LH circuit that makes some animals more or less resilient to stress or anxiogenic environments, for example, or anxiety in general. Because from the miniscope experiments that I showed you, I can tell you that leper cells are inhibited by PFC neurons in a manner that relates to how anxious is the animal, right? But this does not tell me whether prefrontal cortex input is just much stronger in the higher anxiety animals, for example, or whether the leper cells are more sensitive for some reason to this particular input. Yeah. So there are many, many open questions and that need to be answered in this regard. Yeah, I think it's an excellent question, and we need to do more work in order to really address this. And then we have a question by Syed Moise Javed, sorry if I do not pronounce your name correctly, who is curious whether there are any clinically available leptin-based therapies or modulators that are used to treat anxiety-related or metabolic disorders linked to hypothalamic circuits. Yes and no. So there are leptin-based therapies to treat metabolic disorders. They work quite well under conditions where patients have very low leptin levels. So there are human patients who have mutations in leptin, the leptin gene, or in the leptin receptor gene. And they either do not produce enough leptin, or they have difficulty with leptin-based signaling. And these patients can benefit from leptin-based therapies. Yeah, so these are metabolic disorders such as lipodystrophy, for example. And there are these case studies in anorexia nervosa patients that suggest that leptin treatments can also benefit their depressive and emotional symptoms to some degree. Although we do not yet have a clinical study that really would resolve whether leptin-based treatment is actually a good treatment for anorexia nervosa patients, as a systematic study. And yeah, there's one trial in the UK that is currently recruiting, and I'm very curious about the results. But so far, leptin-based treatments are not used to treat neuropsychiatric disorders, and we're trying to do the groundwork to see whether we can make an argument for clinical studies to apply leptin-based treatments to treat neuropsychiatric disorders. Okay, then we have a question by Sumodai Hazra, who wants to know whether the social exposure, I guess from the Optogenic experiments, was done in a novel or a familiar animal. So from the optogenetic experiments where I showed you that the animal had the opportunity to prioritize different stimuli, different social food, water, stimuli. In this case, the animal was familiar. Yeah, we generally use in this first study, very familiar environments and stimuli, that the animals really, really knew. So in some ways, unintentionally, we used very low anxiety environments. And that was possibly very, very lucky, because we see in this later study that we did how strong are the effects of anxiety on leper cells. Okay, and then maybe that would be the last question, or maybe a short question. Let's see. So we have one question by Elsa Volkfock, who wants to know whether leptin's effects on behavior exhibit sex-specific differences, and how we think sex hormones regulate leptin signaling pathway, and what implications does this have for diseases more prevalent in women, such as anorexia nervosa. Yeah, I think that's a very, very good question. And I think we should do more groundwork to really find out what are the sex-specific differences of leptin. You would be surprised as how little we know about sex-specific differences, generally speaking, and especially in relation to leptin mediated effects on behavior. What is known is that the absolute expression, so to speak, or secretion of leptin is very different between males and females. But whether the signaling pathways, so to speak, the neural circuits that are responsible, whether they are different across the sexes is not known. What is known is that leptin does interact with sex hormones. So the secretion of leptin has a particular dynamic across the menstrual cycle in humans and also in rats. That is timed according to the secretion of estradiol and progesterone. Yeah, so this interaction of the hormones in terms of what we can measure in the blood is known. What's the functional implication? We do not yet know. I think these are very important questions that we need to address if we actually find out whether they have implications for diseases that are more prevalent in women, such as anorexia nervosa, as well as depression, for example. Exactly. So then, yeah, we can answer one more question maybe, and then I think we should call it a day. Yeah, maybe just like the last technical question, because I think that's an important clarification by Ines Adam. She wants to know how we actually administered leptin. Was it by an injection or was it orally administered? Yeah, I think that's an important technical clarification we should do. The injection of leptin that we did was via systemic injection. We inject it intraperitoneally. Although leptin has also been directly infused into different brain areas, including in the lateral hypothalamus, in other studies done in other labs. And these were really important studies that showed the importance for leptin in different neuronal circuits, including the lateral hypothalamus for eating behavior. So Chloe, do we want to go on with answering questions? We can if we want.
Yeah, maybe due to time limits, this Q&A part we can end here. And other questions, everyone can contact Anna by email and for the answer. Okay, thank you and very much. Now more than 8,000 articles have been published using fiber photometry and miniscope and of the genetic system, and they have become the necessary technology for the neuroscience research. RWD's fiber photometry and miniscope system can be applied for free trial. If you would like to get more detailed information and try the instrument yourself, we can provide an on-site demo, and welcome to contact us. For those who are selected by Oliver and to answer questions, we will contact you after webinar to confirm the information of the fiber optic cannula delivery. And once again, thanks Dr. Oliver Barnstedt and Dr. Anne Petzold, wonderful course. If you still have any questions, welcome to contact Oliver and by email. We also welcome you to cooperate with Oliver and RWD Lab Science. Finally, thanks everyone joining our webinar. Thank you, Dr. Oliver Barnstedt. Thank you, Dr. Anne Petzold. Thank you, everyone. Wish you have a nice day. See you next time. Bye.
Against all these threats and challenges to life, constant progress of research in life science and tight collaboration in relevant industry chains are essential for achieving better vital quality. Originated in 2002, RWD has been constantly furnishing customers with reliable solutions and services in life science, animal health and clinical medicine, and dedicated for better vital quality within our wits and powers. Our products have been sold to over 100 countries and regions around the globe and won worldwide customer confidence and support. At RWD, we launched 6 solutions in the fields of life sciences, animal health and clinical medicine at the forefront of the world, including animal surgery and modeling solutions, which ensure experiment efficiency through diverse animal disease modeling and model evaluation techniques. In vivo imaging solutions, which provide tangible tech support in fields such as biology, pharmacology, materiology, and translational medicine, among others, by accurately showing blood flow and changes in other indicators of live beings in visualized imaging. Neurosignal research solutions, which are valuable tools for neurocircuit analysis, as well as research into mechanisms for neural system diseases, development of brain-machine intelligence technology, and more. Histopathology solutions, which improve the quality and efficiency of pathological diagnosis while sectioning with high accuracy and efficiency. Cellular and molecular biology research solutions, which advance efficient scientific research through covering all conventional experiments, including cell separation and purification, self-culturing, self-analysis, and molecular biology. Animal diagnosis and treatment solutions, which evaluate diagnostic efficiency and treatment efficacy of surgery with anesthesia, dental services, rehabilitation, and physiotherapy, urinary system diseases, etc. And our assistants, science researchers, have published thousands of papers in a number of world-leading academic journals, including Nature and Science. On top of that, many scientific research institutions, medical institutions, and pet diagnosis and treatment centers have attained their success by leveraging our 6 solutions up to now. We will maintain cooperation and innovation with an open mind, boosting growth within many industries through joint efforts with our partners.
So we've been collaborating and cooperating with RWD for quite some time now, and their self-developed products have helped us in the laboratory maintain accuracy and improve accuracy and help our efficiency. We've used everything from cryostats for sectioning various organs of the body, all the way through to stereotactic surgery solutions to deliver small amounts of virus to very minute regions of the brain. We're really happy with RWD's products, and we're really excited to see what they've got in store, their future products that they have in store for the future, and helping researchers worldwide.
Mein Name ist Dr. Heinrich Koddebusch. Ich bin seit 30 Jahren praktizierender Tierarzt und seit 20 Jahren beschäftigen wir uns schwerpunktmäßig mit der Narkosetechnik in der Kleintierpraxis. Uns Tierärzte treibt das Wohlergehen unserer Patienten an, sowie die ständige Verbesserung ihrer Lebenserwartung und ihrer Lebensqualität. RWD hilft uns dabei mit hochmoderner Beatmungstechnik und ebenso modernen Lösungen für Diagnostik und Therapie. RWDs Hilfe können wir Tierärzte sicherer und effizienter operieren, aber gleichzeitig sorgt RWD auch für eine verbesserte Lebenserwartung. Das Wohlergehen unserer Patienten und die ständige Verbesserung ihrer Lebenserwartung und Lebensdauer treibt uns Tierärzte an.
As a national high-tech enterprise, RWD ascribes great importance to technology research and development. Based on 4 technical platforms, including optical imaging, precision transmission, accurate temperature control, and detection of weak signal, we have a grip on leading core technologies across many fields. Since RWD's founding, the company has invested hundreds of millions on research and development, gaining almost 100 patents, both domestic and international, as a result. In addition to a sound manufacturing base, advanced processing assembly line, and precision detection system, RWD has built an integrated control system that monitors all aspects of operations from research and development to manufacturing. The name RWD comes from the transliteration of the English word reward, meaning payback to society. Reward is not only the original aspiration of the creation of RWD, but also the continuous belief of the company. In 2016, RWD launched the RWD Preclinical Medicine Enlightenment Scholarship to help with the development of research talent in China's preclinical medicine. So far, the scholarship has been awarded to 38 institutions of higher education nationwide. As the Vice President Unit of the Huxley Wildlife Conservation Society, RWD has donated medical equipment and supplies for wildlife relief and the conservation of the natural ecosystem. In 2020, RWD donated clinical medical supplies worth over RMB 1 million to the Giant Panda National Park to help with safeguarding our national treasure. In the future, RWD will continue to contribute its wits and powers to enhance vital quality and help global customers unlock vital mystery and safeguard vital health.